online audience is going to be present as well. And for now, I'm just going to say again, welcome to today's data science seminar. We have Dr. Ariel Rokem here with us and a wonderful speaker. And Ariel will help us with the introductions. And before that, we'd like to do a land acknowledgement. As part of the University of Washington, we acknowledge the Coast Salish peoples of this land, the land which touches the shared waters of all tribes and bands within Suquamish, Tulalip, and Muckleshoot nations. Thank you for your presence here today. And I would like to invite Ariel and Neda to make these introductions. Thank you.
Hi, everyone. Welcome to this eScience data science seminar. We have Dr. Neda Janshad with us here. She's an associate professor of neurology and biomedical engineering at the USC Mark and Mary Stevens Neuroimaging and Informatics Institute at the University of Southern California, where she directs the Laboratory of Brain eScience and is also the associate director of a very large international neuroimaging consortium known as ENIGMA. And in her group's work, she focuses on developing tools and algorithms that assess brain structure, microstructure, connectivity throughout the lifespan, connecting those measurements to all kinds of brain dysfunctions and outcomes for people with mental illnesses, developmental and neurodegenerative disorders, and dementias. And today she'll tell us about multisite neuroimaging analyses.
Thank you so much. And thank you, Dr. Oakham, for having me here today. I'm really excited to start to travel again, and especially come to see you all in Seattle. So yeah, thanks. Today I'll be talking about evolution of multi-site neuroimaging analysis. I tried to keep this talk a little bit more general in terms of data science. So hopefully this will be useful for everyone. Just some standard funding disclosures. A lot of the work that I'll present is actually part of a large-scale consortium, so it's not funding that it necessarily is related to me, but a lot of other groups around the world. So for those of you who might not be working in neuroimaging specifically, the human brain is arguably one of the most important and complex systems that we have yet to understand. So there are a lot of different input sets. We have genetics, we have all sorts of environmental factors, all interact, all individually that interact with brain outcomes, and not just at a single time point, but dynamically throughout life. So it's a huge area for data science. And every decision we make, every outcome has so many input variables that we have to consider if we're trying to model anything related to the brain. For example, in certain cases, an individual might be diagnosed with a neurological or psychiatric disorder, have certain symptoms, and that's not only detrimental to that person necessarily, but to the people around them, to society at large. So it's actually studying the brain actually has so many different components to it in terms of input and output. So this is, hopefully we'll try and get this across in the next few slides. Two examples that I'd like you to hopefully guess are just two very specific outcome variables that we work with in my group. The first one, hopefully you can guess this. In 2019 alone, the estimated cost of this condition was nearly $94 billion in the US. One death every 11 minutes, second leading cause of death for youth aged 10 to 24 in the US, and 10th leading, or one of the top 10 leading cause of death among all age groups, 10 to 64. This condition is seriously considered for nearly 1 out of 5 high school students, and nearly 1 out of 10 high school students are affected by this. Any guesses? Yes. Close. It's actually more specific. So suicide. Suicide, ideation, suicide attempt is one of the biggest problems for youth in the US, and it's costing the United States nearly $100 billion annually. Example number 2, 2018 alone, these were the most recent stats, the estimated costs in the US were nearly $400 billion for care alone. And it's projected to cost $1.1 trillion in about 25 years. 6.5 million Americans are currently living with this condition. And you can see the numbers increase with different age brackets. This number is projected to be nearly 13 million, and some specific outcomes related to this condition include language, reasoning, conscious thought, sensory processing, memory loss, and confusion. And now the NIH has nearly $3.1 billion dedicated for research funding. And this isn't included in the billions that are required for care. Hopefully this is an easier one. Yes? Exactly, yeah. So,
Can you turn on your camera?
oh yeah, sorry. Oh no, I can't. Is that all right? Hide this. So yeah, so Alzheimer's disease is a huge financial burden for all of us, really. So these are just a couple reasons why we are trying to study brain disorders at large. So these two disorders are costing nearly $450 billion in the US alone. So these are huge economic conditions, but they have little in common if you think about them from a neuroscience or a psychiatric perspective. But from a data science perspective, they actually do have a lot in common. We need novel and advanced engineering approaches and data science methodologies to really tackle these problems and explore brain imaging data. So how can computer science, data science, and computational approaches be paired with the biomedical and healthcare sciences to tackle these disorders and so many more? Neuroimaging is just one of many tools, and that's just something that my lab focuses on. So that's what we'll be talking about today. So neuroimaging, we have seen in several published work that with MRI, so this blue line right here, you can actually predict a transition to from normal to impaired cognition much sooner than you can just with clinical observation alone. There are some more invasive methods that are better predictors, but MRI itself is non-invasive, doesn't have any radiation. So it's essentially a lower risk modality. On the other hand, for suicide, for example, there have been some papers published that have found clusters of brain activation that might differentiate individuals who are having suicidal ideations from those that are not. I'll come back to this work too. So there are many types of brain imaging data. So for those of you who might not have worked with imaging data itself, there are different contrasts, there are different modalities, there are different feature sets that you can derive from brain imaging data. And this is all from MRI, or MRI derivatives, I should say. You can look at connectivity profiles, you could look at certain contrasts within various regions of the brain. My group works on a lot of these different modalities and a lot of different features, but this talk will mainly focus on structural brain imaging, looking at features such as the size of certain regions, and this diagram hopefully shows some example of one feature that we can extract. So this movie is going through and extracting the central sulcus of the brain, and you can examine its width, you can examine the depth along the entire length of the sulcus. So this is just an example feature that you can extract from brain data, and you can see how this is just one of very many things that you can extract. More and more papers now, you might have seen, are highlighting these reproducibility or lack of reproducibility issues in neuroimaging. So a lot of neuroimaging studies actually need very large samples to have enough power to detect really anything at all in terms of behavior, and also in regards to genetics. And if we go back to that paper that I showed you on suicide, you see these sample sizes are not large at all. The one that claims to show a predictive pattern of activation differences between ideators and not used a sample size of 17 ideators and 17 controls. And as data scientists, we know that that's probably not going to be reproducible. But that's not, I'm not trying to criticize this paper at all, just saying that we really need to advance what we're doing in the healthcare data sciences to be able to have sample sizes large enough to be reproducible. There are a lot of statistical challenges in big data neuroimaging, and there are some papers that are now showing in order to detect even 1% of the variance, you need thousands of samples. What a lot of my group has worked on is looking at specific genetic markers that might influence brain-related traits. And these genetic markers often explain far less than 1% of the variance in a trait, close to 0.1%. So you could see the sample size needed is actually an order of magnitude larger. You need at least tens of thousands of data points to be able to reliably estimate or find an effect with imaging. So just to give you an idea of what we do with genetics and brain, you could have a genetic study looking at certain cases, let's say Alzheimer's versus controls, and you find positions along the DNA or single nucleotide variants that might influence that disease. One way we can approach the combination of genetics with neuroimaging data is to map those genes or those variants that we know are showing those case control differences onto the brain and see what parts of the brain show variability with respect to that variant. Another thing we can actually do is try and use the brain to look for genetic variants. What are some genetics that might influence certain areas of the brain or one region versus the other? So that's another approach to imaging genetics. And you can do this really with any imaging feature. You can look at the voxel-wise level or every single point, looking at the contrast with these tiny 3D volumes. You can look at connections, and again, look at the many features that we talked about that you can extract from brain imaging data. There are numerous large scale data sets that are available, and they're becoming more and more prevalent. There's a huge open science movement in neuroimaging, and I think that's really added a lot of value to neuroimaging. But one thing that we're doing now is trying to combine those data sets that are now becoming more and more prevalent, like those large scale biobanks that I showed, the Alzheimer's disease neuroimaging initiative, all these existing resources that are becoming public, and these are often huge multi-million-dollar grants that are making this data available. But there's also so many different studies that have been collected on a smaller scale. Let's say you just want to run a study on 20 people or 100 people, and they don't compare to the thousands and thousands of samples that have been collected in large scale biobanks. but maybe there's a way to merge them all together. So that's what that motivated the Enhancing Neuroimaging Genetics through Meta-Analysis Consortium. At the time that we started this consortium, data sets, the largest data set was probably ADNI, which had at that time about 1,000 individuals. Everyone really kept the data to themselves. There wasn't really a much push from funding bodies to make data public. And so now there is, but there didn't used to be at the time where where this consortium was created, and this was about maybe 12 years ago now. So we realized there has to be a way that we can pool existing data together from different universities, from different scientists, that maybe we can't share the actual individual level subject data, but maybe there's something we can do to pool inferences across these data sets and bring them together. So I think of ENIGMA now as a dynamic and collaborative neuroimaging consortium, and it seems to really be organically growing in scale and scope. So I'll talk about that a little bit more. A lot of the sites now, so we have a lot of groups now that are interested in contributing data to a large scale consortium. And a lot of them study rare or uncommon diseases that you might not have a large sample size even in a public population-based sample. Let's say the UK Biobank has something like 50,000 subjects with neuroimaging data, but not all of them, or not many of them, would have a rare genetic variant. Let's say they would not have certain deletions in their chromosomes. So you need to have studies that focus on collecting those individuals if that's what you want to study. So we can pool all this data together, hopefully to answer questions about reliability and generalizability that you might not even be able to get from 50,000 individuals. And those of you who have worked with the UK Biobank know that it's primarily British. So if there's something that affects Americans and not the British, you probably won't find it in the UK Biobank. So now we're aiming to pool all these data sets that there was no coordination between them. There's no coordination on what scanner they'd use to collect, who would the participants be, what the inclusion-exclusion criteria were. Everything is very different. It's very heterogeneous. So we're aiming to deal with really messy data, honestly. And now there are a lot of specific working groups within this consortium that might focus on a particular disease. There are groups that work more on the algorithmic side. I'm more involved in a couple of the neurobiological groups and then a couple of the algorithmic groups. But as you can see here, I'm not going to go through them all, but we do have a lot of variability in the types of disorders that we study and also the types of normal variation that we're looking at. If anyone is interested in how these are organized, there's actually no requirement to share your data if you're going to join one of these working groups. And I think that is something that has motivated a lot more participation. We don't necessarily operate on a centralized level, but a lot of our work is distributed. So if you're interested in participating but are not allowed to share data, you have very sensitive data that must remain in a particular location, you can still join, and we'll have workarounds for that. So there are some people that might have data very specific to one condition and will join only that group. There are folks that have data that might relate to, let's say, Parkinson's disease and suicidal behaviors, because they might have collected information on both, and they could join both groups. So we have a lot of variety within a group and also across groups and some crosstalk as well. Again, I get asked this all the time, like, can I access ENIGMA data? No, because I don't have it. It's really distributed. We're not a data repository. It's more of a consortium. It's a network. Some people do share their data, and maybe they don't have the computational infrastructure to do some of the processing, so they'll share it and have other groups do the processing, but it's not meant to be a data repository. Anyone who's a member can propose a project. Let's say I'm interested in a very specific cognitive score related or motion score related to Parkinson's, and I will ask what Parkinson's groups might have collected data on this score. And I'll put in a proposal. I'll say I'm only interested in this specific neuroimaging feature, this specific cognitive outcome, who has data on what, and what can we do an analysis together. So these projects end up being very collaborative. It's more of an iterative process. People will propose this project. Someone might say, no, that's not a good idea, add this. So things will organically change through the research community. And really everyone in these groups are experts in that particular domain. So the questions get more and more refined and more related to the task at hand. So if you've worked with neuroimaging data, you know this all too well, but imaging acquisitions are different across even within a particular scanner. If you change one parameter, you're going to get a completely different image for the most part. This example is the same person scanned on two different scanners. One is a 1.5 Tesla, one is a 3 Tesla magnet. Same location, a couple weeks apart. You'll notice so much variability between these two that at first glance, you might not realize it's the same person. But in fact, it is. You could see different, obviously, just the grayscale is different. You can see a little bit more detail on this one, not on that screen, but on mine, trust me, there's a little bit more detail. And not only is the imaging different, so not only have the data been collected on different instruments, but oftentimes when you're combining data from these different sites, you're dealing with different populations, as I mentioned, different inclusion-exclusion criteria. And it's not always the same tests that are run. Some groups are interested in a certain battery, while others are interested in others. So one philosophy is that that's actually okay, and that might improve your chances of generalizability and finding an effect that really does generalize across the world. You can have a biomarker where it doesn't matter if you had a particular sequence on a scanner or not, but it actually works across multiple MRI machines. So one way to think about it is that the biomarker has limited clinical utility if it's only good when extracted from one given scanner. And so maybe we can make use of this variability and this diversity in the data set and have features that are more meaningful. So as I mentioned, we don't actually collect the data a lot of times. What we do in this consortium is do a lot of analytical harmonization rather than working with the raw data. Again, it was we're trying to tackle the reproducibility crisis and sort of send out detailed documentation, detailed analytical plans, ensure the same features are collected across these data sets, and the same analysis is run, and also the same quality assurance is conducted. So a couple of things that these are two students in my team, and a lot of our work is looking at the reliability of features, the reliability of methods, the compatibility across, let's say, different versions of software that are commonly used. So when we first started this consortium, the focus was on genetics. As I showed you, it's expanded far beyond genetics, and we're doing a lot of different disorders and a lot of specific conditions. But the first project was screening the genome for variants that might affect the hippocampal volume, because the hippocampus in the brain is really important for so many different conditions. It's affected in numerous conditions. It's the memory center. We see it affected in depression, obviously Alzheimer's disease as well, and many other neuropsychiatric disorders. So this might be a good place to start to screen the genome for markers that might affect the hippocampus. One reason to look into the genetics is because it's a clear biomarker and can be used for drug targets. Like you can target a certain protein for a certain gene, and that might be able to help again have more targeted treatments for such conditions. Anyway, getting back to the hippocampal volume study, a lot of groups had already segmented the hippocampus from their data sets. And so we weren't going to say, do it all over again. Like that's a lot of work for anyone who has tried to manually delineate a specific region within the images. But use whatever you have. We will write the statistical scripts for you. We'll write the quality control scripts. These are the things that you need to look for when you have outliers. Do this, but run these same statistics, and we'll pull the results. So if you're finding an effect in this region and another group is finding an effect in another region, we'll try and in the regions of the genome, we will meta-analyze all the effects together and come up with an overall effect for every single variant along the genome. So that's what we did. Our first paper, the one that I just mentioned, we had a sample size of about 8,000. If you correct for all the variants in the genome, you have about 1 million independent variants. So we set our significance threshold to 5 × 10⁸ to account for those million independent variants. And we had something just below the significance line with the replication sample. It actually was slightly above this Y. So the x-axis is the position along the chromosome. So every single dot that you see is actually a specific genetic variant. The y-axis is -log10 of the p-value. So the higher it is, the more significant it is. But the sample size of 8,000, just shy of significance, kind of made it with replication. Our next iteration, we had a similar paper that looked at more regions, but also included the hippocampus. Now we had more buy-in from different groups around the world. Our N was 13,000. Again, our significance became a little bit stronger, confidence grew. And soon after, we combined efforts with another consortium called CHARGE, and our sample size was 26,000. And you could see how with every repetition of this same analysis, the same statistic that we're running, as the sample size grows, our confidence in our results grows. We had more power to detect this variant. More recently, that same finding that we had in our 2012 paper was also found in another 10,000 individuals of a completely independent sample. This was all collected in the UK Biobank. This was all collected on a single scanner. I think there were close to 10,000 people on this in this study, all from the same first scanner that the UK Biobank used. And you can see that, again, there's this continual strengthening of this effect and the confidence that we have in this effect. And I'll come back to this, but we are now, in terms of the features that we extract, we do a lot of reliability analysis. We work with a lot of data that has been released that include retest samples. So a lot of people might have been scanned 2 weeks apart, 3 months apart, sometimes on the same scanner, sometimes not. Can we extract the same feature? How reliable is it the second time around, or how similar are these metrics? So when we're developing our image analysis protocols, we always tend to ensure that our metrics are reliable. We have detailed documentation. Some of our protocols are containerized. We have harmonized quality assurance metrics. Sometimes you might run some standard software and you forget to look at the output. Hopefully no one in this room does that, but you've worked with people that don't look at the output of their data, you know better. There are protocols that we've developed to common errors of a lot of these pipelines, things to look out for. Like sometimes in these examples, part of the cortex isn't captured. Maybe you don't want to include this region in your analysis, and so forth. Initially, we've worked with groups that have manually quality controlled thousands of these brains, of these images. They've looked for particular errors, and now what we're trying to do is build a model that has learned from these visually assessed images, what the good regions are, what the bad regions are, how to determine whether there's a fail, not just in the overall output, but with specific regions. So there are members in my team actively working on automated quality assurance. And this has gone into using even larger sample sizes. So our latest genome-wide work had about 35,000 MRIs in total. And instead of looking at the hippocampus and subcortical volumes, which I showed before, we're looking at the outer surface, the cortex of the brain. We're looking at the area and the thickness of each of these different colored regions. So we actually did find numerous significant regions within the DNA that affect oftentimes very specific regions of the brain. And we're not pooling individual data sets, we're pooling the results of the individual data sets. So for example, in this forest plot, each line is a cohort or a site that was involved in the study. And this is for one particular genetic variant, how each site showed an effect or maybe didn't show an effect. So you could see a lot of these sites, maybe the effect was right around this dotted line, which is 0, no effect. So you wouldn't be able to see this effect in that particular site. But if you pool the inference from across all these sites, you do have some consensus that there is an effect of this variant on this particular brain structure. So we are doing this in a meta analytic fashion. Again, genetic data is very sensitive, so a lot of people are very uncomfortable sharing that across sites. So we don't enforce it. Data stays where it is. Just the algorithms get distributed and the results come back. So even through this meta analytic procedure, we could still use these same protocols, these same quality assurance metrics, these same feature extraction protocols, not just in the genetics working group, but across all the disorder working groups. And now we can have some overarching view of how these disorders compare to each other in terms of their brain signatures. So we see, for example, schizophrenia shows much stronger effect sizes between schizophrenia cases and controls than, for example, depression. This is major depressive disorder. There's very few regions with a much weaker effect, and certain disorders like 22q deletion syndrome, which is basically a deletion of part of the 22nd chromosome, has what looks to be the opposite effect, where certain areas of the cortex are thicker in the cases than those without the deletion. So you can see these divergent patterns and actually similar patterns across many of these disorders if you do harmonize the analytics and distribute similar protocols. And each one of these studies generally had a couple thousand participants in them. A lot of this we're trying to automate. A lot of the analysis we initially had scripts basically embedded into R scripts that you could call from just a Google sheet. So all really anyone needed was just the key to the row within a Google sheet that was run for one particular study. And you could look into these sheets and you could see the variables that went into a variety of different studies. We actually ran into a little bit of a hiccup with this just recently because R started doing some weird things when reading from Google Sheets, but we're converting it to Python, which doesn't have that issue. So that's for the distributed analytics. There are some groups now that are becoming more and more comfortable with sharing data, especially if it doesn't revolve around genetics. If you're looking at a lot of times older individuals that might have a neurodegenerative disorder, a lot of times that data is actually easier to share. So within our Parkinson's disease group, we were not only able to look at case control differences, but when we pooled the data from all the different sites, and by pooling the data, I mean the individual subject level data, we could split this up and look at different stages of Parkinson's as well. So using the Hoehn-Yahr staging criteria, we could see that progressively within each stage, there are stronger effects in the brain, which you would expect. But because every contributing cohort did not have enough of like a particular stage of the disorder, this was not an analysis that we could do in a distributed manner, but it had to be centralized. Another study I want to highlight, or an aspect of the study that I want to highlight, is when we also have a group looking at the effects of immunosuppression and viral load in people living with HIV infection and how that affects the brain. Now we have a lot of different sites from around the world involved in this working group. We have a lot of groups in the United States, but we also have groups in Thailand and groups in South Africa, where HIV is often is more prevalent, and they are doing larger studies there. Now, interestingly, if you look at the data distribution, almost all the sites within the US were largely male. But if you look at South Africa, it was 95% female. So not only do we have this problem of scanner confounds, because the South Africa site was also the only site that was collected on a 1.5 Tesla magnet, rather than a 3 Tesla higher magnetic power magnet. So we have these competing confounds of differences in sex distribution and also differences in scanner. So now we are actively working on harmonizing raw data by raw data, I mean the images themselves, rather than a lot of harmonization methods now look at the distribution of features and try to do scaling of the distribution parameters, the mean and variance of certain features. But here we're trying to match the style of a particular image. So we're using style-encoding GANs for this, and hopefully this animation does show here as we change the reference image, you can see these different source images change contrast without changing anatomy to match the style of our reference image. Now, for those of you working in diffusion, that's a whole different area that we haven't tackled yet. We also are doing a lot to try and categorize the different data sets. I won't go into this, but my group is very involved in the informatics of this and trying to structure the data, looking at the metadata, trying to really derive similarities in terms of concepts, because a lot of groups might collect a similar variable but not call it the same thing. For example, a lot of groups will label something sex, the other one would call it gender. So, but those obviously have, in the case of these neuroimaging studies, they are meant to capture the same concept or similar concepts. So we want to create these ontologies that try and capture as much of the similarities as possible. A lot of these data sets were collected many years ago too, so a lot of the concepts might have meant something and then sort of might not mean the same thing anymore. So there's a lot of variability in the data sets that are involved in ENIGMA, and we're trying to put some structure around the actual terms. There are some groups putting structure on the neuroimaging derived features, and we're working with them as well. And that actually allows us to incorporate this idea of continuous analysis, where if you have, going back to that original genome-wide association study that I showed, where you have one result with a sample size of 8,000, and you can increase confidence as you increase the sample size. What if you could do that automatically? So if you have your metadata all labeled in a certain way, you might be able to find new data that exists that can be used to answer a hypothesis or answer a question that you've asked already. And can you incorporate that data automatically into this into the same pipeline that you've already, the analysis have already been run, the scripts have already been distributed? Can you incorporate that in and continuously refine your confidence? So we're trying to do a lot of the infrastructure behind sort of a continuous analysis model for neuroimaging. Right now it's, we're calling it NeuroDisc. And right now it's focused on genetic results for the cortical surface area work that I showed. So here, one of my students, Alice, is working on, let's say, automatically incorporating more data. I think one of these sites was, so the UK Biobank was not in this one, was not in the original implementation, but then when you added that in this one, you see that the confidence was, I'll find a little bit, sorry, the slide is very blurry, but not only can you look at these forest plots and meta-analyze the data, but you can also relate it to different features of the cohorts. For example, the mean age if you have that captured in the metadata, you have information about the cohort, you could see whether your effect of these particular data sets might relate to another property of that data set. So for example, we regressed mean age on the effect size derived from the data set, and you can see some weak relationship between the age of the sample and the effect that it showed on the brain. So there are numerous questions that still remain with brain imaging, and hopefully, if you haven't worked with brain imaging data, this will motivate you to start looking into it, because there are so many unanswered questions. A lot of times we need to really merge the brain imaging data with other data types. There are lots of data sets that are now collecting follow-up scans. How reliable are some of these features in the follow-up scan? How reliable are the effects? How well do some of these predictive models work with longitudinal outcomes? There's a lot of work we're trying to do there. Like let's say instead of predicting what your face looks like from age 40 to 65, predict what your brain looks like. So we have this predictive model for brain aging now. And this image right here is showing our results of pooling data sets from around the world that have looked at suicidal ideation and suicidal attempt. We are still seeing in over 1,000 individuals, rather than a much smaller sample size, we're still seeing very weak effects with neuroimaging alone. So we really do need to boost the data types and incorporate multimodal information in a lot of these brain conditions, and being able to effectively predict outcomes. So yeah, thank you for listening. Thank you to those that tuned in online as well. And I'd like to thank a lot of people, hundreds of investigators that have really contributed to ENIGMA, and a lot of the folks that have highlighted their work here as well. So thank you. Any questions?
So the ENIGMA project seems to be neat, and there's all the levels variance in data. And something that occurs to me is that, especially with the informatics kind of looking, it seems like there's an opportunity there to answer questions about how those variances actually affect the features of the data. So, for example, we all know that different scanners have features in the scan, but it's really hard to get an answer to the question: what's the consequences on my baseline search for the scanner versus that's seen scan? That's a very trivial example, but I'm curious to know if you're trying to do any of that work.
That's absolutely something we're looking into, especially with this. Oh, sure. So he asked whether with these large scale sample sizes, when you're pulling data from all these diverse sources, you could see if something like scanner manufacturer affects the outcome. And that is absolutely something we're looking into now. We've actually been asked that question in a lot of our papers. So for example, in our diffusion study of looking at case control differences with schizophrenia, it doesn't matter if you have more than 30 directions if you're just doing an FA analysis. So we took the data, we split it up, looked at those that had less than 30 directions and groups that had more, and we found very similar effects. But it really, any case might be different. There might be a feature that Philips isn't picking up very well, and we're trying to prevent that earlier by looking at this test-retest reliability across multiple sites, so that scanner hardware would not be as much of a contributor to differences, but perhaps the distribution of the population might be. So so again, something that we were trying to do is looking at just, does the age of the cohort matter? So yeah, I mean, now we can really start when we have like hundreds of groups participating, we can try and tease apart whether certain features are more important. Yes.
I have a question about this idea of the continuous analysis. So the way you presented it here, continuous analysis, every time new data comes in, it automatically gets incorporated into the analysis.
Hopefully, yes. We're working on it still.
And the question is, what do you do when you have a new when the analysis method improves?
That's a great question too. You that's always an issue. So we are we've tried to answer some of that very specifically. So we looked into, let's say, free surfer segmentation. There's a version upgrade. How much of a difference do these upgrades in analysis tools make? There are certainly differences. There's no answer to what you should do with your data yet. Should you upgrade everything? Should you run everything the old way? Should you do half and half? Should you just keep going? But I do think that as we have more and more data, and maybe, so the data sets, the data quality will also improve with time, and so will the analysis methods. So hopefully, eventually we can use the older analysis with the older data, sort of leave it, have it done, and then update the newer data sets as we go along. But again, I'm just speculating. That's a great question, and we're trying to understand that more too.
Any other questions? Okay, if not, I would like to thank our speakers today, and please join us next time for the data science seminar. Thank you.